In 2010, a tiny fragment of a pinky finger was unearthed in a Siberian cave. Geneticists expected the bone to belong to a Neanderthal, but the DNA told a different story. Researchers sequenced a full, high-quality genome from that single finger bone. The genome revealed an entirely new branch of human evolution, one nobody saw coming. They named these mysterious cousins the Denisovans after Denisova Cave in Siberia where the finger was found. The discovery raised a huge question. How could an entire species remain hidden for so long? Where are their bones? Almost nothing has ever surfaced, just that one finger and a few teeth. One bold theory says we may have already found them and just didn't realize it. Not in Siberia, but in tropical Java, Indonesia, thousands of miles away. Not one bone, but 12 skulls, sitting in museums for decades, labeled as something else. That's the wild claim at the heart of this investigation, and it's backed by some surprising evidence. Since 2010, Denisovan research has exploded. Jawbones, teeth, even a half-Neanderthal hybrid child have been found. Then, a jawbone from a cave in China, analysed not by DNA but by ancient proteins, was identified as Denisovan, the first fossil of their kind found outside Siberia. At Denisova Cave itself, they found something even stranger, the bone of a child with one Neanderthal parent and one Denisovan parent, the only known direct hybrid from the ancient world. Denisovan DNA didn't just stay in Siberia, it turned up inside living people today, especially in New Guinea and Australia, where up to 4% of the genome comes from this ghost population. Even China's massive dragon man skull, with its huge brain case and thick brow, was floated as a possible Denisovan, but without DNA, it's just a very good guess. To make sense of all this DNA, I called population geneticist Joe Teixeira, who joined me from his home office, looking like he was about to commentate on a cricket match. But to understand where Denisovans fit, we have to rewind to a pivotal chapter in human prehistory, the out-of-Africa migration. Modern humans left Africa sometime between 55 and 100,000 years ago, a single group that would eventually spread across the entire globe. Soon after leaving Africa, this group met Neanderthals, and instead of fighting, they had children together. That encounter left a permanent mark. Neanderthal DNA from that single meeting survives in every non-African population today, a ghost from a brief romance. But as humans kept spreading across Asia, they met another group, the Denisovans, and absorbed them into their communities too, adding another layer to our genetic tapestry. Different regions absorbed wildly different amounts of this ancient DNA. Some populations in island Southeast Asia carry up to 4% Denisovan, while others have almost none. Curious, I tested my own DNA and found out I'm more Neanderthal than 72% of people. My wife, to my dismay, beat me 95%. The results came back, and I learned I am more Neanderthal than 72% of people tested. But then my Vietnamese wife took the same test and scored even higher, 95%. She beat me again. This proves that admixture levels vary enormously by region and ancestry. Not everyone carries the same ancient ghost in their blood. One region stands out far above all others for Denisovan DNA, and it's not Siberia. Island Southeast Asia, New Guinea and Australia carry the highest proportions of Denisovan ancestry of any modern humans. About 4% of their genome traces back to Denisovans, a sizable chunk from a ghost population. Compare that to roughly 2% Neanderthal DNA in most Europeans, it's double the ancient inheritance. Could that really mean Denisovans once lived in that region, mixing with the ancestors of people there today? Two competing explanations exist, and the answer isn't obvious, it's a genuine scientific puzzle. Maybe the mixing happened right there in island Southeast Asia, or maybe it happened somewhere else entirely, and later populations obscured the trail. Every population there from Papua New Guinea to the Philippines descends from the same African migration as everyone else. No evidence suggests any separate group colonized Australia independently, they all came from that same out-of-Africa wave. This lets scientists compare the DNA of Australians and Papuans to the DNA of neighbouring groups in mainland Asia, looking for the same Denisovan signal. Enter the Andaman Islanders. Their ancestors colonized the Andaman Islands soon after the first migration out of Africa, and then remained in relative isolation for thousands of years. If the Denisovan mixing happened before this group split off, the Andaman Islanders should carry the same high level of Denisovan ancestry as people in New Guinea and Australia. They don't. The Andaman Islanders show noticeably lower Denisovan ancestry. The signal is much weaker than in the populations of island Southeast Asia and Sahul. That points to the mixing happening closer to island Southeast Asia, after the Andaman Islanders had already split off and gone their own way. A third clue deepens the puzzle. The Denisovan DNA found in island Southeast Asia looks genetically distinct from the Denisovan genome recovered from Siberia's Denisova Cave, meaning at least two separate Denisovan populations, one in Siberia, one in the south, each contributed their own distinct ancestry to modern humans. Based on all this evidence, the shared migration, the isolated Andaman Islanders lacking the signal, and the distinct southern DNA, geneticists now feel confident that a Denisovan-like population lived in island Southeast Asia, But confidence from DNA isn't the same as a body. Genetics can point to a ghost population, but to really know the Denisovans, we need their bones. Time to search the fossil record to see if any of the ancient skulls and bones already sitting in museum drawers might actually belong to these elusive Denisovans. Island Southeast Asia holds one of the richest collections of fossil hominins on Earth, a jumble of ancient humans that lived and died across these tropical islands. Meet Sangaran 17, a skull from Java dating to between 1 and 1.3 million years old, a classic example of Homo erectus, one of the first large-brained hominins. Sangaran 17, found on Java and dating to well over a million years old, is the textbook Homo erectus fossil. Big-brained, tool-making, wildly successful, and built like a beast. Homo erectus spread across Eurasia and Africa for eons, producing sophisticated hand axes and controlling fire. They were the first true global human species. Erectus may have survived on Java until around 100,000 years ago, meaning they were still there when modern humans first reached the region. But nearby, on the island of Flores, sits a hominin that breaks all the rules, a tiny species barely a meter tall. This specimen, called LB1, lived around 70,000 years ago and belongs to the species Homo floresiensis, nicknamed the Hobbit. Its place on the human family tree stays fiercely debated. Some see it as a dwarfed erectus, others as something far more ancient. One theory, Homo erectus got stranded on Flores and over generations shrank through island dwarfism, the same process that shrinks elephants on islands. Another theory, Floresiensis descended from a separate, earlier migration out of Africa by a form of early Homo, like Homo habilis, that predates erectus entirely. Further north, in the Philippines, another newly identified hominin species emerges, Homo luzonensis, named after the island of Luzon. Its fossils date back at least 50 to 67,000 years, placing it squarely in the same time window as modern humans arriving in the region. Its evolutionary position, too, remains genuinely uncertain. Some features look primitive, like Australopithecines, others surprisingly modern. All these lineages, Erectus, Floresiensis, Luzonensis, split from our own lineage millions of years back, making them what geneticists call super-archaic hominins. Every one of these groups, Homo erectus, Homo floresiensis, Homo luzonensis, still lived in island Southeast Asia when modern humans arrived. Modern humans reached Australia by around 65,000 years ago, judging from finds like the Majed Babi rock shelter. They reached Sumatra by around 73,000 years ago, according to the dating of a single human tooth found there. That timing overlaps directly with the last known appearances of Homo erectus, Homo floresiensis, and Homo luzonensis. It's very likely that modern humans crossed paths with all of these groups when they arrived. That raises a new question: did modern humans interbreed with these older, more archaic groups too? Researchers, including Juro Teixeira's team, set out to test for that exact kind of ancient genetic mixing. Quick note, the term archaic sounds negative, but it's just a label for older lineages that split from our main line long ago. Every human alive today, every single one, carries some archaic DNA from Neanderthals, Denisovans, or both. The real obstacle for testing super-archaic admixture: no reference genome exists for Homo erectus, Floresiensis, or Luzonensis. Unlike Neanderthals and Denisovans, there is no ancient DNA sample from any of these groups to compare against modern genomes. So researchers model the exact number of mutations expected if two populations had split millions of years ago and then met again. If modern humans had interbred with Homo erectus or Homo floresiensis, their descendants' genomes should contain stretches of DNA that look deeply ancient and highly divergent. Joao's team didn't just look at genomes from island Southeast Asia, they scanned samples from populations across the entire globe, and they found them, small portions of the human genome carrying highly divergent sequences present in every modern population tested. Exciting until you realize evolution is messy. These divergent stretches could come from a super archaic ancestor or something else entirely. So the team ran a check. If these divergent sequences were really from a super archaic, they should not correlate with known Denisovan or Neanderthal ancestry. They found exactly that. The more Denisovan ancestry a population carried, the more of these divergent sequences they had. That correlation means the simplest explanation: these divergent regions are just undetected Denisovan DNA, not a separate super archaic species. The team's best estimate for any true super-archaic contribution in modern genomes? At most 1/10 of 1%. Even that tiny fraction shows how far science has come, looking for ghost ancestors in a sliver of our DNA. Verdict from the genetics: a clear, strong Denisovan signal in islands Southeast Asia, but no confirmed evidence of mixing with a separate super-archaic group. That leaves exactly two possibilities on the table for the fossils of island Southeast Asia. One, true Denisovan fossils are still out there, buried somewhere in the islands of Indonesia, New Guinea, or Australia, waiting to be found, or the fossils already sitting in museum drawers have simply been mislabeled the whole time. So let's revisit a set of 12 skulls pulled from the banks of the Solo River on Java back in 1931. That expedition was led by Dutch researchers who were retracing the footsteps of a famous earlier discovery, Eugène Dubois's 1891 find on the same river. Dubois had unearthed a skull cap and thigh bone that he first called Pithecanthropus erectus, upright ape man, now known as Homo erectus. Near the village of Ngandong in 1931, the new team hit something extraordinary. 12 fossil skulls emerged from that single excavation, an almost unheard of haul in paleoanthropology. Modern dating of the sediment around those skulls places them between roughly 117,000 and 108,000 years old. Officially, these are labelled Homo erectus, but is that label actually correct? A provocative question has started to circulate. Could these 12 skulls actually be misclassified Denisovans? One complication, the timing looks awkward at first glance. Our own lineage, Homo sapiens, split from the Neanderthal Denisovan line around 600,000 years ago, but the famous Sangiran 17 skull, also from Java, belonged to a population that lived over 1 million years ago, long before that split even happened. On its face, that makes those older fossils clearly not Denisovans. They split from our lineage way too early. But that logic only holds if nothing changed for a million years, and that's an enormous assumption. We already know one disruption happened, modern humans arrived and replaced the local populations. Nothing rules out this Java lineage later mixing with newcomers, the same way Denisovans mixed with our ancestors elsewhere. Humans themselves mixed with multiple archaic groups across their migrations. It wasn't a one-time event. Even the common ancestor of Neanderthals and Denisovans mixed with an older archaic species around 600,000 years ago. So assuming total isolation for 1 million years on Java is a stretch, especially when we know mixing happened everywhere else. That opens the door back up for the Ngandong skulls, the 12 fossils found along the Solo River. That reopens real doubt about whether Homo erectus is even the right label for them. Scientists have argued for a century over how to classify them. They never quite fit the boxes. Their brains were larger and rounder, their skull bones notably thinner, features that don't match classic Homo erectus. A 2010 paper even proposed a separate name, Homo soloensis, after the Solo River where they were found. That paper arguing over whether the Ngandong skulls were Homo erectus, Homo sapiens, or something else entirely, was published in 2010, just 1 month before the first Denisovan DNA paper dropped. It makes you wonder, if that classification paper had come out just a little later, would the Denisovans have factored into the debate? To get another perspective, the host reached out to John Hawkes, a well-respected anthropologist based in America. Hawkes replied that he'd thought about the Ngandong material as possible Denisovans and found points both in favour and against the idea. But Hawkes emphasised that without genetic or biochemical evidence from the remains themselves, we may never know for sure. One objection, the Ngandong skulls closely resemble earlier Homo erectus features, which seems to argue against them being Denisovans. But that doesn't rule them out. Shared features could simply reflect later admixture between Denisovans and local Homo erectus populations. Curiosity naturally turns to another famous skull from the region, Dragon Man, discovered in China and dated to around 146,000 years ago. The two skulls share some traits. Both are robust with thick brow ridges, but differ in other clear ways, like overall shape and brain size. But remember, the Denisovan population that contributed to island Southeast Asian genomes was genetically distinct from the Siberian Denisovan from Denisova Cave. No one actually knows how that genetic gap between the two Denisovan populations would have manifested in their physical appearance. And here's the humbling truth, we only have one full Denisovan genome to work with, so we have no idea how variable that population was across its range. Neither of these objections is strong enough to rule the theory out. Without fossil DNA from the Ngandong skulls, John Hawkes agrees the question stays genuinely open. A real gap divides what genetics implies and what the fossil record confirms. Only ancient DNA from actual bones can close that gap. Hope now rests on future testing of the Dragon Man skull and the Ngandong remains. That testing could confirm this theory, disprove it, or reveal something entirely new. One striking pattern. Homo erectus and Denisovans share the same geography. Both apparently occupied the very same stretch of ancient Asia. That overlap forces a rethink of what Denisovan even means. Maybe species boundaries were never as clean as textbooks suggest. Gene flow between similar groups can blur any strict dividing line. It is compared to arguing over where dogs end and wolves begin. The case for the Ngandong skulls being Denisovan rests on 3 pillars. Strong genetics pointing to a Denisovan-like population in island Southeast Asia, a rich fossil record in that very region, and the simple fact that no one has yet extracted DNA from these bones to settle it. What makes the Ngandong skulls so interesting is that they don't look like classic Homo erectus. They have larger, rounder brains and thinner skull bones, a strange mix of archaic and modern features that has left paleoanthropologists debating their classification for decades. That ambiguity has puzzled researchers for roughly 100 years. Ever since the first Ngandong skull was dug up in 1931, scientists have gone back and forth. Is this Homo erectus? Archaic Homo sapiens? Something else entirely. Verdict. These 12 skulls have a genuinely strong shot at being Denisovan. They come from the right place, the right time, and they have the right kind of morphological ambiguity to fit the genetic picture. If they are Denisovan, they may represent the very last surviving Denisovan population known to science, a group that persisted on Java until roughly 108,000 years ago, long after their Siberian cousins had vanished. Ruling out Homo soloensis entirely would still take a leap of faith. The fossils are so hard to classify that dismissing them as just another Homo erectus population ignores the very real possibility that they are something more. This raises a simpler question: which is more likely? That a completely unknown fossil population of Denisovans is still hidden somewhere in the jungles of island Southeast Asia, or that the fossils already sitting in museum drawers have simply been mislabeled. Misclassification looks like the far more likely explanation here. We already know that paleoanthropologists have been arguing about these skulls for a century. It wouldn't be the first time a fossil was reclassified after new techniques came along. The original 1932 paper describing the Ngandong skulls already compared them directly to Neanderthals. The Dutch paleontologist who named them Gustav Heinrich Ralph von Koenigswald wrote that they may represent a stage of human development equivalent to that of the Neanderthal race. A century of scientific debate has circled back to nearly that same idea. After decades of being labelled as evolved Homo erectus, the Ngandong fossils are once again being considered as close relatives of Neanderthals, and by extension, Denisovans. These skulls may be close relatives of both Neanderthals and us, the Denisovan branch of the human family tree, living out their final days on the island of Java, while modern humans spread across the region. That thread ties Ngandong directly back to the vanished Denisovans. The same fossils that once puzzled scientists as an evolutionary dead end may actually be the missing link between the genetics and the bones. If this idea holds up, the missing Denisovans were never missing at all. They were hiding in plain sight in the fossil collections we already had. They may have sat in museum drawers for nearly a century, mislabeled as Homo erectus, just waiting for the right question to be asked. That single pinky bone from Denisova Cave, which started this entire scientific hunt, may now have 12 possible siblings waiting in the collections of Java. Nothing is proven yet, but the evidence, genetic and fossil, is pointing somewhere new, somewhere that connects a tiny Siberian cave to the tropical islands of Southeast Asia. If you want more real, evidence-based journeys through human origins, subscribe and hit the bell. The next chapter of this story is already being written. From one finger bone to 12 skulls, the Denisovans may finally be found, not in a remote Siberian cave, but in the collections we've had all along.